Hello everyone, welcome to my YouTube channel Biotech Talk. Today we are going to discuss about production of antibiotics. तो guys इस लेक्चर में हम डिस्कस करने वाले हैं कि एंटीबायोटिक्स का प्रोडक्शन किस तरीके से होता है. तो फर्स्ट ऑफ ऑल गाइस वी आर गोइंग टू अंडरस्टैंड व्हाट इज एंटीबायोटिक्स. कंपाउंड दैट किल और इनहिबिट द ग्रोथ ऑफ अदर ऑर्गेनिज. ऐसे कंपाउंड जो किल कर देते हैं, इनहिबिट कर देते हैं अदर माइक्रो ऑर्गेनिज की ग्रोथ को, उनको हम क्या बोलते हैं? एंटीबायोटिक्स. मोस्ट एंटीबायोटिक्स आर प्रोड्यूस बाय फ्लामेंटस फंगी और एसिटोनोमाइसिटिस. इनकी हेल्प से ये जो है मोस्टली एंटीबायोटिक्स प्रोड्यूस होते हैं. दे आर ड्राइव फ्रॉम द स्पेशल माइक्रो ऑर्गेनिज्म और अदर लिविंग सिस्टम एंड आर प्रोड्यूस ऑन इंडस्ट्रियल स्केल यूजिंग फर्मेंटेशन प्रोसेस. इनको हम इंडस्ट्रियल स्केल पर प्रोड्यूस करते हैं फर्मेंटेशन प्रोसेस का यूज़ करके. टुडे ओवर 10,000 एंटीबायोटिक सब्सटेंस हैज़ बीन रिपोर्टेड. आज की तारीख में 10,000 एंटीबायोटिक सब्सटेंस जो है रिपोर्टेड हैं. Antibiotics are produced by fermentation. Any large-scale microbial process occurring with or without air is called fermentation. This process may take a few days to obtain in acceptable amount of product. Antibiotics produce is done by batch process. Antibiotics का production हम batch process से भी करते हैं. Next one is properties of microorganisms. Properties of useful industrial microbes including grow repeatedly on the large scale in expensive medium, produce desired product quickly, should be non-pathogenic or amenable to genetic modification. Product produced by microorganisms like microbial cells, enzyme, antibodies, steroid, alkaloid, food additives, and ethanol, citric acid, etc. Are these products produced by microorganisms? Primary metabolite are the intermediate and product of metabolism. Primary metabolites, these metabolites which we require for the growth and maintenance of cellular function are called primary metabolite. ऐसे metabolite जो growth के लिए और cellular function के maintenance के लिए जरूरी है उनको हम बोलते हैं प्राइमरी मेटाबोलाइट. प्राइमरी मेटाबोलाइट आर फॉर्म ड्यूरिंग द ग्रोथ फेज. एग्जाम्पल अमीनो एसिड, विटामिन, कार्बोहाइड्रेट, लिपिड, न्यूक्लिक एसिड एंड एंजाइम. सेकेंडरी मेटाबोलाइट वो मेटाबोलाइट होते हैं जो ग्रोथ और सेलुलर फंक्शंस के लिए के मेंटेनेंस के लिए ज़रूरी नहीं होती. एट द एंड प्रोडक्ट ऑफ प्राइमरी मेटाबोलाइट. सेकेंडरी मेटाबोलाइट जो है एंड प्रोडक्ट होते हैं प्राइमरी मेटाबोलाइट के. सेकेंडरी मेटाबोलाइट आर फॉर्म नियर द स्टेशनरी फेज ऑफ ग्रोथ. Drug, toxin, steroid, and polymeric substance like rubber are the some example of secondary metabolite. These are the example of secondary metabolite. Primary metabolite and secondary metabolite. The first diagram is secondary primary metabolite and second one is secondary metabolite. Primary metabolite are made during the growth. Example protein, enzyme, nucleic acid, ethanol and lactic etc. These are the example of primary metabolite. Their production rate follow the growth curve. Secondary metabolites are not produced during the normal growth. Their production usually started after the normal growth phase. Not all microbes produce secondary metabolite. Production of antibiotics. The mass production of antibiotics began during World War II with spectromycin and penicillium. Now, most antibiotics are produced by stasis fermentation, in which strain of microorganisms produced high yield are grown under optimal conditions. Production of antibiotics can be done by three methods. First one is natural microbial production, जिसमें हम यूज़ करते हैं फर्मेंटेशन टेक्नोलॉजी का. एग्जांपल पेनिसिलियम. सेकंड वन इज सेमी सिंथेटिक प्रोडक्शन, जिसमें हम पोस्ट प्रोडक्शन मॉडिफिकेशन करते हैं नेचुरल एंटीबायोटिक्स. एग्जांपल एम्पीसिलियन. थर्ड वन इज सिंथेटिक प्रोडक्शन ऑफ एंटीबायोटिक मेड सिंथेटिकली इन द लैब. एग्जांपल गुआनिलीन. Fermentation technology. The source of microorganisms grew in the large container 100,000 to 150,000 liter or more containing liquid growth medium. Oxygen concentration like temperature, pH, nutrient level must be optimum. As antibiotics are the secondary metabolite, the population size must be controlled very carefully to ensure that maximum yield is obtained before the cell die. Fermentation process require following. First one is a pure culture of chosen organism is sufficient quantity. जो प्योर कल्चर आपने चूज़ किया है वो सफिशेंट क्वांटिटी में होना चाहिए, स्टरलाइज होना चाहिए, केयरफुली कंपोज मीडियम फॉर ग्रोथ ऑफ द ऑर्गेनिज्म. A seed fermentation is minimal model production fermentation to develop inoculation to initiate the process to main fermentation. main ferment process को initiate करने के लिए. The production fermentation, the functional large model equipment like drawing the culture medium seedly state, cell separation, collection of cell-free supernatant, production of purification, effluent treatment, constitute of upstream and downstream of the fermentation process. Strain used for the production. Species are the often genetically modified to yield maximum amount of antibiotics. फर्स्ट वन इज म्यूटेशन म्यूटेशन को इंट्रोड्यूस करने के लिए म्यूटाजेनिस को इंट्रोड्यूस करेंगे अल्ट्रासाउंड रेडिएशन यूवी रेडिएशन एक्सरे इन सब का यूज़ करेंगे सिलेक्शन एंड फर्दर रिप्रोडक्शन ऑफ हायर yielding strain can be rise yield by 20 fold or more another technique used to increasing the yield like recombinant DNA टेक्नोलॉजी का यूज़ करेंगे raw material the compound that make the fermentation broth are primary raw material required for the antibiotic production the broth is aqueous solution made up of all the ingredients required for growth of the microorganisms typically it contain carbon source हमारे हो सकते हैं molasses soya meal acetic acid alcohol hydrocarbon these material are needed for the food source for the organism nitrogen is another necessary compound it is metabolic cycle of the organism ammonium salt is typically used other element, tract element needed for the proper growth, antibiotic producing microorganisms like phosphorus, sulfur, magnesium, zinc, antiforming agent to be preforming during fermentation like lard oil, octaconol. Steps in production. First, the organ that make antibiotics must be identified. Desired microorganisms must then be isolated. Then organisms must be grown on the scale large enough to allow purification and chemical analysis of antibiotics. The antibiotic is tested against a wide variety of bacterial species. It is important that sterile condition it is maintained throughout the manufacturing process because contamination by foreign microbes. First one is starting a culture. Before fermentation process, the desired microbe must be isolated and number must be increased by many times. Start a culture from the sample is previously isolated organism is created in lab. A sample of the organism is transferred to agar containing plate. The inertial culture is then transferred to say flask containing nutrient necessary for growth. A suspension is formed which then transferred to seed tank for further growth. This is the diagram of fermentation. Seed tanks are still tanks designed to provide the ideal environment for microorganisms. The seed tanks are equipped with mix which is mixed growth medium with microbe and pump lever sterilized filtrate air. After about 24 to 28 hours, the material in the seed tank is transferred to the primary fermentation. Second one is fermentation. The fermentation tank is larger version of the seed tank, which is able to hold about 30,000 gallons. Microorganisms are allowed to grow and multiply during the process that excrete large quantity of desired antibiotics. Tanks are cooled to keep temperature between 23 to 72 degrees Celsius. Constantly aggregated and continuously steam of sterilized air and pumped into it. Antiforming agents are periodically added. Third one is isolation and purification. After 3 to 5 days, maximum amount of antibiotic will be produced. The isolation process can begin. The isolation depends on the specific antibiotic production. This is the diagram of how to isolation of microorganisms and purification, refining, then packaging and shipping. Water soluble antibiotics. Antibiotics compound that are water soluble or ion exchange method is used for purification. These compounds first separated from the waste organic material in the broth, are sent through the equipment which is separate other water soluble compound. Oil soluble antibiotics. The solvent extraction method is used for the isolation of oil soluble or organic antibiotic. The broth is treated with organic solvent such as butyl acetate or methyl isobutylbutane which is dissolved in the antibiotics. The dissolved antibiotic is recovered using various organic chemical methods. At the end of this step is purified powder from the antibiotic is often which can be further refined into the different product type. Quality control is a great importance in production of antibiotic. Antibiotics के production में quality control great importance role play करती है. It is involved fermentation process में भी वो involve रहती है. Step must be taken to ensure that absolutely no contamination is introduced at any point during antibiotic production. During manufacturing, the quality of all compound is checked. at the regular basis. Frequently check the condition of microorganisms in culture. Industrial production in penicillium. Penicillium was first important commercial product to produce by aerobic and submerged fermentation से इसको produce किया गया था. Penicillium cryogenesis species is used. Production penicillium produced by fungus Penicillium cryogenesis which is require lactose, other sugar and source of nitrogen in case of yeast extract in the medium of grow well. Like all antibiotics, penicillium is secondary metabolized